Afternoon. I'm going to describe some of the work we've been doing, and right away I've got my jumping thumb on. This is my requisite slide of aflatoxin contamination of corn. This is what we're trying to prevent through biological control. And what you're looking at are Canidia, mycelia, this fungus on the corn ear. And when A. flavus grows, when it gets to be about a day old, and it comes in contact with the substrate-air interface, it starts making canidiophoros. It's developmentally regimented to do that. These emerge, a vesicle forms, little fingers come out up here with nuclei in them. The nuclei divide and go into another little cell, canidium, and those continue to divide, making chains of canidia. The youngest here, the oldest up here at the top. And as this is going on, at about the same time, the aflatoxin synthesis pathway is kicking on. And it's beginning to make aflatoxin and secrete it into whatever system it's growing in. And what I want to talk about today are three areas. I want to talk about research we've done on the mechanism of biocontrol, which we think is intraspecific aflatoxin inhibition. And we think that's mediated by a physical interaction, a touch inhibition. Non-toxigenic and toxigenic need to touch. The second thing is population biology of A. flavus in Louisiana cornfields with regard to specificity of infection, what strains infect and what don't, and the involvement perhaps of mating type in infection. And finally, what we're interested in are the biocontrol results that we've gotten using atoxigenic strains. And what makes our approach a little different, I think, is that we have the idea that multiple strains are better. And the reason for that will become, I hope, become clear to you when we talk more about the mechanism. This paper in 2003 by Wicklow in Peoria demonstrated a very important principle. He looked at aflatoxin production by growing toxigenic and non-toxigenic together in the suspended disc. And this is the suspended disc. You put 9,000 canidia of A. flavus in there in various ratios of toxigenic to non-toxigenic. A little water in the bottom. And let them grow for 5 days. Took this out, extracted the aflatoxin, and looked at the result. And this is what Wicklow found. If he had 100%, all 9,000 were toxic producers, he got this. If all 9,000 weren't, he got this. If 80% were toxigenic and 20% were non-toxigenic, he didn't get a 20% reduction, he got a 70% reduction. If it was 60 toxigenic to 40 non-toxigenic, he didn't get a 40% reduction, he got nearly a 90% reduction. So clearly, there's not a one-to-one situation where it's just consuming carbon and pooping out aflatoxin. There's more to it than that. We use that technique to screen some 41 isolates from corn kernels, and we paired them with a single toxigenic strain at 50-50 or 80 toxigenic to 20 non-toxigenic. And when we did that, grew in the suspended disc for 5 days, looked at toxin production. The blue ones at both ratios, I think there's 8 of those, didn't make any toxin. These isolates inhibited 53 from making toxin. The purple ones were highly inhibitory. So out of that 41, 12 of them were highly inhibitory, and this is how they broke down by strain, or vegetative compatibility group. There were 3 major VCGs, we call them A, B, and C. These are the members of A, B, C, and there were 2 singletons over here. But the important thing is, isolate 53 was the same VCG as these non-toxin producers, and this is kind of forbidden, but it happens. So this is evidence that within the same VCG, you get inhibition of aflatoxin inhibition. You might think they just fuse and continue to make, but that's not what happens. So, after doing that, and the reason we did that was to look at potential biocontrol strains, those that are intraspecifically inhibiting aflatoxin production. We did an experiment using this, co-opted somebody else's technique, Janisovich at West Virginia, I believe. They had a system where they looked at biocontrol, nutrient-based, an apple pathogen problem, penicillium. They put here in this filter insert plate well method. This is a cylinder with the filter in the bottom and a well that's put in. So if they put the pathogen here, the yeast antagonist here, and apple juice in there, and let the apple juice and the metabolites go back and forth through that filter, what they showed was that that prevented penicillium from developing and was the reason for biocontrol. When we did that with isolate 53, the toxigenic, and isolate 51, which we found in suspended disc culture growing together, were inhibitory. In this case, we got toxin. When we did the experiment slightly different way, when we mixed them, 1:1, 53, 51 in glucose salts medium, put half in the insert, half in the well, we got no toxin. Reconstituted what happened in the suspended disc culture. We interpreted that as meaning that touching or physical interaction was important for downregulating toxin synthesis. So if this touching or physical interaction is important, allowing this fungus to pass through the membrane should result in toxin inhibition. The diameter of the conidian hyphae is about 3.5 to 7 microns. So this was an experiment that was done. If we put the toxigenic one in alone, 3 filter sizes, we got 400 to 600 parts per billion of aflatoxin. If we mix them together and put them in the well and in the insert, we got 0 to 10 parts per billion. If we separated them into the 2 compartments using a 3 micron or a 0.4 micron filter, this is not significantly different from this. There was no inhibition. Down here, these are holes you could drive a truck through. Here they're essentially mixed, and these aren't any different than these. 12 micrometer one is about halfway between. When they're starting to touch, they're starting to inhibit. What we've also showed here is that there's specificity in this touch inhibition. Just because an isolate inhibits strain A doesn't mean it will inhibit strain B. So there's specificity, and this means that if this is the biocontrol mechanism and there are multiple toxigenic strains in the field, you've got to use biocontrols which will inhibit all the sets of toxigenic strains that you have out there. And that's the basis for our trying to use multiple strains for biocontrol. This shows the specificity that I just mentioned. This was our isolate 53. This is at 51. The reason we became aware of this, we were trying to look at this touching, and we had a GFP strain. And when we put our GFP strain with 51, it didn't inhibit. These are some of the other ones that were in that initial work that I mentioned. This is 21882, K49. But you can see there are other strains which do inhibit 70S GFP. And it looks very similar to the sequence strain 3357. These two are fairly similar. This one's quite different. Now, I don't know how many different specificity profiles there are out there, but presumably there could be many of them. So our conclusions from this mechanism work was that inhibitory ability is BCG independent. Competition for nutrients or soluble inhibitor signal molecules can't explain this. We think that touch or physical interaction is needed, and this inactivates a known signaling pathway to prevent toxin synthesis. We showed their specificity, and again, that means we need multiple isolates to deploy. We showed the window of sensitivity to inhibition. They have to be touched in the first 24 hours of the toxigenic strain's growth. The inhibitory one is always capable of inhibiting, and those results are published in PLOS 1. So you think this may be kind of a strange situation, touching and fungi. Well, there's a little bit of precedent for this in another aspergillus. This German group in 2009 showed that in order for this secondary metabolite to be produced by nidulins, it had to be touched or grown in close physical contact with the streptomyces in soil. They went on to show that this requires histone acetylation to turn on that secondary metabolite pathway. So this is an example of an induction of a secondary metabolite pathway by touching. Ours is a repression of a secondary metabolite pathway by touching. Also, you should note that histone acetylation is important and occurs in the cluster when it's turned on as well. So there's perhaps something here about preventing histone acetylation is what's involved in the mechanism of not allowing toxin to be synthesized. Now I want to go to the second part, population biology. Peter alluded to this earlier. It's important to know what's out there in the field in order to know what you have to control. And we did a survey of 11 fields where we sampled soil and kernels in August. And the results of that were that we had approximately, this represents 300 soil isolates from those 11 fields. This represents about 600 isolates from corn kernels from those fields. If we determine the vegetative compatibility groups or strains, this approximately 100 separated into 16 different strains or VCGs. These out here are not characterized, but they're not these 16, they're others. When we looked at corn, we found only 6 VCGs in corn, 16 in soil. So clearly, all of the strains in the soil don't go to corn, and the frequency of this one in the soil is much, much less than the frequency it has in corn. And we'll talk more about that later. But the point of this is we think this indicates that there are strains in the soil which go to corn and strains in the soil which don't go to corn. When we did this work, we also looked at aflatoxin production by these strains on rice, growing them just on rice, extracting toxins, see how much they made. I just want to focus your attention on the first two. That's VCG1 and 4. That's the blue one here, 10% of the isolates from kernels. This is about 80%. That was a surprise. 80% of them were this single VCG. So if we focus on aflatoxin production by those two, VCG1, 4 isolates from 2 locations, mean aflatoxin 3.3 parts per billion. 483 isolates from all 11 locations, average 4.5. Less than 5 parts per billion by that strain. Look at VCG4, the blue one. 5 isolates from 2 locations, 56 isolates from 11 locations. Over 20,000 parts per billion. That suggests to us that VCG1, highly infective in corn, low toxin producer, may be a natural native biocontrol strain out there in the field doing what I'm trying, what AMCOE's paying me to try to do. So we're looking at VCG1 isolates as potential biocontrol strains. It's clear that it infects corn relatively easily, and that's another spin-off of this is, I started on this in '98 when we had our severe outbreak, and we've had many years since then when the environmental conditions, the heat, the drought, have been as conducive as '98, yet we haven't had a severe outbreak. Why? Well, it's possible that these high toxin-infected strains are not indigenous to fields where the corn is infected. That is, they fly in from elsewhere. They may be aliens. Why do I say that? Well, 2 of the 11 fields we found in soil, 1 and 4, and we didn't do an exhaustive isolation from soil, but it suggests that they get around, the ones that get into corn are more, higher frequency of isolation of them from corn than from soil. So that may be a reason. Rust of wheat flies in every year from Mexico. Their spores are 10 times bigger than this. There's no reason why we can't have an aflatoxin or aspergillus flyway that comes in as the corn ripens, and we're exploring the possibility of these being aliens, not soil-borne, not in the soil in which the corns grow. The other 3 points I'd like to make is to have a bad year, why we hadn't had a bad year. We need a conducive, stressful environment, which I said we've had. We need the highly infected, high toxin producing strains. And we need an absence or no VCG1 indigenous natural biocontrol strains. I think that's what may be going on, at least in that year in our fields. What I didn't show you in that previous slide, that VCG1 and 4 were both mating type 1-2. There's 2 mating types here. And the question is it VCG of 1 and 4, are there genes there that make it go into corn, or is it mating type 1-2, or both that increase infection? So we did an experiment in which we put an isolate of VCG1 from corn, an isolate of VGC4 from corn, VCG8, which we found only in the soil, and one of the unknown VCGs from the soil, which we presumably find in corn. Put them on wheat in the field at silking, let the corn mature, harvested, isolated from the kernels on each adjacent row, and asked, of the 20 isolates adjacent here, how many were VCG1? 1 of 20, 0 of 20 here, 0 of 20, 0 of 20, 1 of 20, 1 of 20, 2 of 20. So VCG here didn't have much effect on infection. The question is, what was the mating type of all those strains? And to make a long story short, 95% of them were mating type 1-2. At the other end of the field, we sprayed the same 4 VCGs on the silks, or a nip mutant of the 4 VCGs on silks, weighted, harvested, isolated from the kernels, 20 isolates. In this case, this, remember, was our 80% infector. Half of them, when sprayed on the silks, 10 of 20 were VCG1. The mutant, 4 of 20. So 14 of 40 were on there. So clearly there must be something in VCG1 that facilitates corn infection. The other 3, not any different than when we put it between the row on inoculated wheat seed. Again, 95% BC, mating type 1-2. So, one of the things in our proposal that we propose to do is to trap spores, Canidia in the field, and several applications for this. This is an ionic spore trap which one of my colleagues developed. It sucks air in under this hood, puts it down a channel here which has electrical discharge, ionizing what goes through there. And there's an SEM stub that's charged, and the particulates in that flow stick to that stub. So we put that out in the cornfield for about a month. This shows the stub that's removed. We put it into a sterile tube, take it back to the lab, vortex it in a little surfactant, and plate out what's on there. When we did that, we sampled over essentially late July through most of August. We washed the spores from 51 stubs, plated them on AFPA selected media, got 764 colonies. We randomly picked 103 to determine mating type, extracted DNA from 103, performed multiplex PCR mating type assay, ran the gels. 3 were mating type 1-1, 100, or mating type 1-2. But we find that in the kernels, we find that flying. We found that in 11 locations around Louisiana from the population biology study. So it looks like in Louisiana, mating type 1-2 prevails, and it looks like it may be doing something to enhance infection of corn. And if I wanted to be a biocontrol agent, I wanted to get in the corn, and I wanted to touch that incoming toxigenic one, I'd want to be mating type 1-2. So we checked Aflatoxin, AF36, K49, Tex98, and these are some of ours that we selected. I'll show you in a moment more about those. All of these are mating type 1-2. It's a gene that turns on other genes, which allows mating. 1-1 turns on another set of genes, which allows mating. And it's our hypothesis that 1-2 induced genes have a role in facilitating corn infection, and 1-1 induced genes do not. It's a beautiful hypothesis. Unfortunately, this summer knowing there was corn infection in the Midwest, asked colleagues to send me corn from infected areas, and I got one response from Bob Bellum at Brownstown Research Center, University of Illinois in southeastern Illinois. He sent us 9 hybrids from 3 locations. These are the Pioneer hybrids from the Research Center, Macoupin County near there, another Pioneer, Franklin County, a couple of DeKalbs. From those years, we isolated 574 colonies. We picked 90 to test. And this time we didn't get the highly skewed 1-2 ratio. We got roughly 40 to 50 and 2 that didn't amplify. I don't know what's going on here. I don't know if Louisiana is in a 1-2 hole or whether that's the true situation in Louisiana. We need to do more in other locations and see the importance of mating type in infection. What about the biocontrol results? Well, in order to select an effective biocontrol, we took the approach that we found from that population study. We took 10 of our highest producing toxigenic strains, and we paired them individually, ATOX1 with all individually with these 10, ATOX2 with those 10, and summed up the total toxin produced. This is the total toxin produced by the 10 alone, and this is the profile you get from each of these atoxigenics. What you need to note is 17, 19, 21, 22. This is 21, 882, K49, Tex98, Aflatoxin, or AF36, excuse me. So these 4 here that we detected, we thought would be good biocontrol agents, and we went ahead to try to use them. And the way we do the experiments is we coat wheat seed in this case, although lately we've used barley, some use sorghum. We coat them with the conidia, throw them out in the field. If they get wet, they germinate, make those conidia that I showed you earlier. They blow up and infect. We also seed the field after applying this with a toxic isolate. And when we did that in 2010, this is what we got. This was a control toxin alone. Aflatoxin didn't do much, Aflatoxin didn't do much, our ATOX-17 didn't do much, ATOX-19 didn't do much, ATOX-21 a little better, but not very good. But when we put it with 17 and Aflatoxin together, they're better than either alone. 19 and Aflatoxin together are better than either alone. ATOX-21 and Aflatoxin are better than either alone. We did that again in 2011. Aflatoxin, ATOX-17. This is ATOX-17 plus Aflatoxin. 19 plus Aflatoxin. This time we used 51 and 51 plus Aflatoxin. 51 by itself looked pretty good in this example. What we're suggesting here is we need to cover the spectrum of inhibition of the toxigenic strains that are out there. This is 2012, 17, 19, 51, 3. These are a couple new ones from the VCG1 test. 3, 10, and Aflatoxin. Here's Aflatoxin 17. Looks pretty good, but 17 alone looks pretty good. Aflatoxin 19, Aflatoxin 51. 3 and 10. So in most of these tests, it's the combination that is better than the individual application. And I was interested to hear Peter say that in Africa they're using 4 strains. One of the other things that is new last year was Syngenta came out with a sprayable formulation of Aflatoxin. And this was an experiment we did for AMCOE at Arkansas. Bert Blum did, and Tom Allen did at Mississippi State. And what we saw here is from the Louisiana location. This was Tox 4 sprayed on. First what we did, we sprayed Aflatoxin, and a week later came back and sprayed Tox 4, and then at the same time we sprayed plots with Tox 4 alone and a combination of the two. What you see here, I think, is a pretty good effect of Aflatoxin in either sequence of application to controlling aflatoxin. Bert's results were, if I can get to them, were very similar, except if he put it on early, he got an even better protection than putting them on together. But again, both looked effective. Tom Allen's didn't get planted until very late. It was treated, we received the kernels, we assayed them, and we found no aflatoxin in any of them. It was a late test, and there was no usable data from that. Another experiment that we did was looking at the timing of application. The V5, V10 are the tassel stage. And at 2 rates of the sprayable formulation, these are the parts per billion. The thing that stands out is the V10 stage at the high spray rate, 11 parts per billion. There is an explanation for this which suggests that the V10 stage, the V10 is where the ear eventually emerges. If you spray that area, you douse that area with inoculum, the ear comes through there and it's right in the neighborhood, and that may be an explanation of why it's effective. This is an experiment I wish I had done if I could get my thumb under control. This was done by Hamed Abbas at Stoneville. 2 years, 2 locations. And I've shown you this earlier. This is pin bar inoculation of ears with various ratios of non-toxigenic to toxigenic. This looks exactly like Wicklow's suspended disc work, showing the inhibition. I think we showed the touch inhibition is important. I'm going to claim that this is more evidence again that what's going on here is a touch inhibition effect in controlling aflatoxin. Dorner, in an early report, he was the grandfather of Aflagard. He stated biological control of aflatoxin contamination is not associated with reduction of propagules in the treated soil. The competitiveness of the strains in the process of colonizing and hyper-defecting and becoming established is a vital aspect of the success. And this is the summary slide. What I think I've tried to show you today is what we call parasitic biocontrol. And others have referred to this as competitive exclusion. I'm going to call it competitive inclusion. You need both the toxigenic and the non-toxigenic together in this infection court to touch in the first day of the toxigenic isolate's growth. When they do that, it's down regulation of aflatoxin if the specificities are right. Saprophytic biocontrol is another component. Competitive exclusion in this case could refer to the biocontrol occupying the organic debris in the soil and out competing in the soil. It would provide sustainability and would be epidemiologically important because it would be there to make those spores go up and infect or protect. So I think there's at least 2 components to this. I need to acknowledge those that did the work, the girls in the lab, Chung-Lee, Archana, Rebecca, and Kathy, the Louisiana Soybean and Grain Research and Promotion Boards, Agenda, the National Corn Growers, AMCOE, and the LSU Ag Center for their support and for the help of Bert and Tom. And if you have any questions, I'd be happy to try and answer.
Questions?
Do you think that he's talking about maybe not having the, I remember your term about the, maybe in the Midwest, there hadn't been that.
You're talking about mating type?
Yeah, that particular mating type. Do you reckon that, what made it produce or not produce? Because probably it was in the soil anyway.
Well, backing up a minute, what we found in Louisiana in the soil is roughly 1 to 1. And people in North Carolina look, it's roughly 1 to 1 in the soil is what you'd expect for a normal native population in the soil. So what made it so strange to us was the skewed way it was in corn. And we have a number of incidences over a number of years, and we've been looking at isolates from kernels. They all seem to fit that pattern. But for some reason, the Illinois one, it doesn't work up there that way. Either I'm in a 1-2 hole, or although from sampling from the soil would indicate that it's roughly 50-50. So I don't know if I answered your question, but
I still don't know why it didn't. If it's
Why?
that way in the soil, why did it not produce that way on the
well, what we showed from the population biology study was just the frequency in the soil does not reflect the frequency in the kernel. So that's part of the results here, that it's important to know what's out there that gets into the corn and what strains can inhibit that. And that's why I was trying to make that point about BCG that you were talking about. What's the population like over the whole corn growing area? If we do that, that VCG4 strain, incidentally, that we found in 10% was at all 6 Monsanto locations previous year. It's the only one that occurred at all 6 locations, so it clearly gets around or flies in or jumps out of the soil or whatever it does. So, any other questions? Thank you.